with a keen understanding of the system by which blood carries oxygen, nutrients, and other materials throughout the body. While researchers in other areas of science were embracing the trend toward breaking things down into the smallest units, the physiologists understood that there was no minimizing what happens in the body. Even the simple act of standing up involves a series of changes in blood flow, blood pressure, nerve response, and heart rate, all important. This awareness, they believed, enhanced their view of the scientific shift underway. The march toward decoding the body's genetic script had been steady and painstaking. In 1953, James Watson and Francis Crick, relatively obscure researchers at the time, discovered DNA's double helix structure, an advance that enhanced the understanding of how human inheritance worked. In 1977, the Americans Walter Gilbert and Alan Maxam and the Englishman Frederick Sanger discovered two different ways to read the sequence, to identify which of the four chemical bases, adenine, guanine, cytosine, or thymine, A, G, C, or T, lies at a specific point on each rung of the double helix. The entire set of instructions still seemed too vast to read, some 3.2 billion pairs of letters. Then, in the mid-1980s, Sanger's technique was automated, and hope emerged that the entire human sequence could be mapped. When the U.S. government launched the Human Genome Project in 1990, announcing that for the next 15 years it would provide an estimated $200 million a year for the effort, young scientists steered their careers toward the great question of the day: DNA. The new field brimmed with possibility. If researchers could solve the mystery of how genes translate heredity into chemicals that course through the body, it might be possible to control the process. Here might lie the power to drastically improve the length and quality of human life. The scientists on the beach came well prepared to meet the challenges of the new era. Two of them had worked for leaders in the fast-growing field. After receiving their schooling in the Midwest, Jacob and Krieger had both moved to Boston for postdoctoral fellowships under Victor Zhao, a cardiologist who would later serve as the first editor of a journal conceived by Cowley called Physiological Genomics. Zhao would go on to become president of the National Academy of Sciences Institute of Medicine. Under his tutelage, the young fellows had begun learning the language and techniques of genomics as the science took shape before their eyes. While in Zhao's lab, Jacob also undertook a second fellowship, this one with Eric Lander of the Whitehead Institute for Biomedical Research at MIT. Lander, a mathematician turned geneticist, had been one of the key figures in convincing the U.S. Congress to commit billions of dollars to the Genome Project. He would be the lead author on the landmark 2001 paper in the journal Nature that presented a first draft of the sequence, an early benchmark against which other sequences would be measured. It was clear in 1993, 8 years before that draft was presented, that this project would be the crucial event in biology in the last half of the 20th century, arguably as significant to humanity as putting a man on the moon. Many researchers in physiology and other disciplines were defensive, critical of the vast amount of resources being devoted to the genome. They worried that all of the money going into one area would mean that there would be less for their own work. The project, they said, did not represent real science. It turned the centuries-old scientific method on its head. Researchers would be searching a massive data set for answers rather than developing a hypothesis and testing it. But Cowley and his young protégés didn't see it that way. They believed that this effort to draft the human blueprint would fuel research and discoveries for years to come. None of it seemed difficult on that beautiful day in La Jolla. The afternoons slipped